Greetings and welcome to the Professor Kareem Ainsley Encounter. In this space, we'll be discussing matters concerning medical laboratory science, clinical laboratory science, biomedical laboratory science, and laboratory medicine. If you or anyone you know is interested in this topic, please feel free to share this information with them. The aim of this training session is to prepare the student and those in the profession with information as to different areas of specialty, especially if they are considering to sit the ASCP examination. During the process of our discourse, we will be covering the different target areas for the examination, which include but not limited to immunohematology, chemistry, clinical chemistry we're speaking about, hematology, immunology and serology, laboratory management, and other areas as deemed fitting. But for today's discourse, we'll be covering the immunohematology section, which is commonly referred to as blood banking. This will be our entry in the preparation of the board certification examination for the ASCP, American Society of Clinical Pathologists. Today, we'll be looking at fundamental concepts concerning the red blood cells and the platelets preservation. We'll be throwing back a little history and then look at some current trends taking place. This experience should give you an idea as to the teaching and delivery style so that you can decide if this style suits your learning needs. And if so, we hope to see you on the journey as we seek to complete all the core areas as we prepare together. As we go through, I want you to understand that this is not for the purpose of memorization per se. It is for you to understand the concepts so that you can have a basic appreciation to be able to solve problems independently based on the knowledge that you would have already garnered. But if you're new to this space, this will give you an idea as to what is expected in the field of laboratory medicine, clinical laboratory science, or medical technology. Our objective for today is to go through the overview of what the transfusion medicine is all about, describe some biological anomalies and biological consideration for red blood cell survival pre and post transfusion. We're going to be defining some hemoglobin concepts. We will be looking at the hemoglobin-oxygen dissociation curve. We'll be explaining some additives that are used, FDA guidelines, and quite a few important foundational concepts so that you can be prepared when we get a little deeper into the information. So without further hesitation, let us embark on the journey together. As it concerns blood banking and blood in essence, people have always been fascinated with blood. Ancient Egyptian bathed in it, yes, that is true. Aristocrats drank it. Authors and playwrights use it as theme. And modern humanity, as we know it, transfuse it. Order to an efficient, safe, and uncomplicated transfusion has been a difficult one, and that is exactly where we are heading. But the process did not come without its crucibles, its trials and errors. So the story is told in the year 1492 that blood was taken from three young men and given to a stricken Pope Innocentia VII in the hopes of curing him. Unfortunately, all four died. These young men are important. They were not just regular young men. They were actually slaves that the Pope had serving him, and they had to sacrifice their lives for the purpose of science, not by choice, but under duress, under force. However, the story continues as this marks the advent of what we now appreciate as blood transfusion. Although the outcome of this event was unsatisfactory, it was the first time that blood transfusion was recorded in history. It doesn't mean that it had never been undertaken or practiced. It's just that it was the first time that it had been recorded. As time progressed, it would have been realized that clotting was a principal obstacle to overcoming the challenges with the transfusion process. Attempts to find a non-toxic anticoagulant began in 1869 when Braxton Hicks recommended sodium phosphate. This could have been considered the first example of blood preservation research. It was Carl Landstern in the 1901 who discovered actually the ABO blood group as we know it or as was recorded. This discovery also displayed and indicated to us that there are different reactions that occur when there is incompatibility with the blood and blood products that are transfused. So Carl went on to win a Nobel Prize in the 20th century. Then it did not stop there. Next came devices designed for performing transfusion. With every idea, over time it is expanded. And the first successful contraction was designed by Edward E. Lingerman. He carried out vein-to-vein transfusion of blood by using multiple syringes and special cannulas for puncturing the vein through the skin. However, this time-consuming, complicated procedure required many skilled assistants. It was not until Unger designed his syringe valve apparatus that a transfusion from donor to patient by an unassisted physician became practical. So yes, we used to have the physicians who were doing the blood withdrawal techniques. But even before that, we had barbers who were actually the originators of the bloodletting process. And before that, we had the priests who were given authority to do the bloodletting and to do all manner of ritualistic practices. But as it relates to modern blood bank and blood banking procedures, there was an unprecedented accomplishment in the whole area of blood transfusion, which was achieved in the 1914 when Huston reported the use of sodium citrate as an anticoagulant solution for the transfusion. So we had Braxton Hicks, who recommended sodium phosphate in 1869, and in the year 1914, we had Hudson, who reportedly used sodium citrate as an anticoagulant solution for transfusion. Later, in 1915, Lewis Law determined the minimum amount of citrate needed for the anticoagulant and demonstrated its non-toxicity in small amounts. So with these advents and development, transfusion became more practical and safer. In the years that follow, in 1916, glucose was evaluated as a necessary preservative with the introduction by Ross and Turner with citric dextrose solution. The truth be told, the function of glucose in red blood cell metabolism was not understood until 1930s. Therefore, the common practice of using glucose in the preservative solution was delayed. But World War II stimulated blood preservation research, and then we had the pioneer work of Dr. Charles Drew on developing techniques in blood transfusion and blood preservation, which led to the establishment of the widespread system of blood banking. And this was accomplished in February 1941. As we move down the timeline in history, in 1942, Louis and Mollison of England introduced the formula for preservative acid citrate dextrose, ACD. So with this intervention, then in the 1947, there was the issue of the Journal of Clinical Investigation, in which this preservative was published, and this sparked the commonplace of blood preservative, and then blood transfusion became commonplace. With the daily occurrence of transfusion, it led to the discovery of numerous blood group systems, and antibody identification surged to the forefront as sophisticated techniques were used to develop and deduce what was causing what reaction. So yes, we have come to the realization and accepted the norm that the ABO blood group is not the only blood group that exists. But as we progress, we will dive deeper into the other blood groups that exist. It is stated that in 1957, Gibson introduced an improved preservative solution called citrate phosphate dextrose, CPD. So we moved from ACD in 1943, and by 1957 we have CPD. This was said to be less acidic and eventually replaced the ACD as the standard preservative used for blood storage. So now that they have blood and they are storing it, in the past, a single unit of whole blood could serve only one patient. That is just such a waste. With component therapy, however, one unit may be used for multiple transfusion. Today, in our healthcare system, the healthcare providers can select the specific component for their patient particular need without risking inherent hazard of using the whole blood transfusion. Healthcare providers can transfuse only the required fraction in the concentrated form, decreasing the probability of overloading the circulatory system. And this is an advance that has taken us from waste and adding more variables to a transfusion in a state where we can now reduce unlikely variables or unnecessary variables to ensure the efficacy of the blood products and the safety to the patient. It is estimated by the AABB, formerly the American Association of Blood Banking, that 6.8 million volunteers donate blood every year. And this is based on a 2015 National Blood Collection and Utilization Survey. And approximately 12.6 million units of red blood cell were collected, around 11.4 million were transfused. Studies have shown that it is estimated that 1 in 3 people will need blood at some point in their life. These units are donated by fewer than 10% of a healthy population. And when we speak about the population, we're speaking about the American society who are eligible to donate every year. Traditionally, the amount of whole blood in a unit of blood has been 450 mils plus or minus 10% of the blood 1 point. More recently, 500 mils plus or minus 10% of blood is being collected. These units are being collected from donors with a minimum hematocrit of 38%. Modified plastic collection systems are used when collecting 500 mils of blood. With the volume of anticoagulant preservative solution being increased from 63 mils to 70 mils. When you're collecting 450 mils plus or minus 10%, the normal volume of anticoagulant that is already in the bag is about 63 mils. But when you're collecting 500 mils plus or minus 10%, the normal volume of anticoagulant is 70 mils. The total blood volume of most adults is 10 to 12 points. Donors can replenish the fluid lost from the 1 point donated in 24 hours. The donor red cells are replaced within 1 to 2 months after donation. A volunteer donor can donate whole blood every 8 weeks. So we have to remember this. Every 8 weeks, a volunteer blood donor can donate blood. So when the blood is donated, the unit of whole blood that is collected can be separated into 3 components. So we have the packed cell, we have the platelets, and we have the plasma. In recent years, less whole blood has been used to prepare platelets because of an increased utilization of apheresis platelets. Hence, many units are converted only into red blood cell and plasma. The plasma can then be converted into cryoprecipitate to a clotting factor concentrate that is rich in fibrinogen. So now let us recap. So the whole blood that is collected from an individual can be broken down into three areas of separation. We have the packed cells, we have the platelets, and we have the plasma. The plasma can then be broken down into cryoprecipitation or cryoprecipitate, which is rich in fibrinogen. So if persons have specific clotting factor deficiency, then cryoprecipitate would have been a suitable alternative instead of the transfusion of plasma. A unit of whole blood prepared red blood cell can be stored for 21 to 42 days, depending on the anticoagulant preservative solution used when the whole blood is collected and whether a preserving solution is added to separate the red blood cell. We should just bring to your knowledge and attention that donated blood is free. However, there is a cost associated with the collection, the testing, the processing, the storage, the shipping of these components. If you are considering blood donation, there are three main areas that we need to consider. So let us now look at the donor process. The donor process has three predominant steps. One is the educational reading material, two, a donor health history questionnaire, and three, an abbreviated physical examination. In step one, the educational material is material from the AABB, the pamphlet giving information of risk associated with infection disease transmitted by blood transfusion, including symptoms and signs of HIV, is given to each prospective donor to read. Step 2, the donor health questionnaire, which is a uniform donor history questionnaire designed to ask questions that protects the health of both the donor and the recipient. This is given to every single donor. The health history questionnaire is used to identify donors who have been exposed to diseases that can be transmitted in the blood. Example variants of West Nile virus, malaria, Babesia, or Chagas disease, etc. Now to step 3. The abbreviated physical exam for donor includes the blood pressure, the pulse, the temperature reading, the hemoglobin or hematocrit level, and an inspection of the arm for skin lesion, rashes, etc. These are necessary procedures to protect both the donor and the person who will be receiving the donated unit. This also sometimes eliminates the need for wasting donation bags and testing apparatus and material, providing that the pre-screening procedures have been failed. So when it comes on to blood banking, immunohematology, the area of focus in terms of cell are the red blood cells and also the platelets. We do mention the white blood cells only in situation where we have to do some irradiation. However, the red blood cells are our main focus alongside the platelets, and that will be our area of focus during this segment of our study. When we move on to hematology, we will focus more in depth on white blood cell and its morphology. Three areas of red blood cell biology are critical for normal erythrocyte survival and function. Yes, the red blood cells are considered erythrocytes. Number 1, the normal chemical composition and structure of the red blood cell membrane. So if there are membrane defects, then that would impair the ability of the blood cell to slide through the veins and may cause them to be unable to carry enough oxygen as required. Number 2, the hemoglobin structure and function. If the hemoglobin structure is compromised, they are limited, they are somewhat deformed, then it will not be able to hold on to the iron component necessary to carry the required oxygen. And if it is not able to carry the required oxygen, then this individual may have a state of anemia. Then we have the red blood cell metabolism, number 3. And if the metabolism is out, then there has to be some compensatory mechanism in order to restabilize what is happening. Defects in any or all of these areas will result in fewer red blood cell survival than the normal 120 days in circulation. So now we know that blood cells, the erythrocyte, has a 120-day normal circulation time in the body. And after circulation has been completed, the blood cell is sequestered and it is broken down into reusable component and then rebuilt from within the body. So let us just look at the red blood cell membrane once again. The red blood cell membrane represents a semipermeable lipid bilayer supported by a mesh-like protein cytoskeletal structure. A phospholipid, the main lipid component of the membrane, they are arranged in bilayer structure, comprising the framework in which globular protein traverse and move. The proteins that extend from the outer surface and span the entire membrane to the inner cytoplasmic side of the red blood cell are termed the integral membrane protein. Beneath the lipid bilayer, a second class of membrane proteins are called the peripheral protein, and it is limited to the cytoplasmic surface of the membrane forming the red blood cell cytoskeleton. So the membrane of the red blood cell is important for optimal functionality. When we have a compromised membrane, there is an equal relationship with a compromised ability for the cell to perform its duties as intended. The biochemical composition of the red blood cell membrane is approximately 52% protein, 40% lipid, and 8% carbohydrates. The normal chemical composition and the structural arrangement and molecular interaction of the erythrocyte membranes are critical to the normal length of the RBC survival in circulation, which is 120 days. Let us examine the concept of deformability. When we speak of deformability, we basically speak of red blood cell flexibility, its ability to be deformable and permeable. The loss of the adenosine triphosphate, that's ATP, the energy level, leads to a decrease in the phosphorylation of the spectrin and in turn a loss of membrane deformability. So let us just try to imagine this mentally. The loss of the energy, the ATP, leads to the decrease in the phosphorylation of the spectrin. So the energy decreases, the phosphorylation mechanism does not have enough power to function as it needs to. And then this loss in functionality in turn causes the membrane not to be able to flex and morph. Hence, we have the loss in the membrane deformability. An accumulation or increase in the deposition of membrane calcium also results or causes an increase in membrane rigidity and loss of pliability. So we have to remember this as well. Calcium also results in an increase in the rigidity and loss of pliability. These cells are at a marked disadvantage when they pass through the small 3 to 5 micrometer in diameter of the sinusoidal orifice of the spleen. This is the organ that functions in the extravascular sequestration and removal of the age, damaged, or less deformable red blood cell or fragments of their membrane. The loss of the red blood cell membrane is exemplified by the formation of spherocytes. And these are cells with a reduced surface to volume ratio, and bite cells in which the removal of a portion of the membrane has left a permanent indentation in the remainder of the cell. Let us move on to permeability. The permeability property of the red blood cell membrane and the active red blood cell cation transport prevents colloid hemolysis and controls the volume of the RBCs. So we are seeing that any abnormality that increases the permeability or alters the cation transport may decrease red blood cell survival. So let us revisit what does that mean. The red blood cell is freely permeable to water and anions. Chloride and bicarbonate can traverse the membrane in less than a second. It is speculated that this massive exchange of iron occur through a large number of exchange channels located in the red blood cell membrane. The red blood cell membrane is relatively impermeable to cations such as sodium and potassium. Important to know that the erythrocyte or the red blood cell intracellular to extracellular ratio of sodium and potassium are 1 to 12 and 25 to 1 respectively. Intracellular is 1 to 12, extracellular is 12 plus 12 plus the 1 that is there. So that equals 25, and then we just flip it around, 25 to 1. So small to big, big to small. Current donor screening tests for infection. The tests, we're going to just mention the tests and the date they were required. So syphilis was required since 1950s, and we remember the Tuskegee situation. The Tuskegee situation, in which it was a public health crisis and a disregard for humanity. Then we have hepatitis B surface antigen, which was required in 1971. Then we have the core antibodies for hepatitis B, which was required in 1986. Then we had hepatitis C, which was required in 1990. Then we had HIV virus, that is human immunodeficiency virus antibodies 1 and 2, required in 1992. Then we have human T-cell lymphotrophic virus antibodies, that's anti-HTLV 1 and 2, which was required in 1997. Then we had a human immunodeficiency virus, HIV-1, that was required in 1990. Then hepatitis C virus, required in 1999. Then we had West Nile virus, required in 2004. Then we had trypanosoma cruzi antibody, that's anti-T cruzi, that was required in 2007. Then we had hepatitis B virus, HBV, required in 2009. Then we have a Babes microti antibody that was required in 2012, and we had Zika virus required in 2016. The requirements were based on the location of the individual, the geography, and the exposure to the particular ailment. At this time, let us look at the metabolic pathway of the red blood cell. We understand that the red blood cell was designed for the purpose of carrying oxygen to the tissues and also to carry carbon dioxide outside of the body. So in the natural design, the red blood cell does not consume that which it is carrying. The red blood cell metabolic pathway that produces ATP are mainly anaerobic because the function of the red blood cell is to deliver oxygen and not to consume it. Because the nature of the erythrocyte has no nucleus, and there is no mitochondrial apparatus for oxidative metabolism, energy must be generated almost exclusively through the breakdown of glucose. So, red blood cell metabolism can be divided into the anaerobic glycolytic pathway and 3 auxiliary pathways that serve to maintain the structure and function of the hemoglobin. Glycolysis generates about 90% of the ATP needed by the red blood cell, and approximately 10% is provided by the pentose phosphate pathway. The methyl hemoglobin reductase pathway is another important pathway of the red blood cell mechanism, and a defect can affect the red blood cell post transfusion survival and function. Another pathway that is critical to red blood cell function is the Lubering-Rappaport shunt. This pathway permits the accumulation of an important red blood cell organic phosphate, which is 2,3-diphosphoglycerate or 2,3-DPG. The amount of 2,3-DPG found within the red blood cell has a significant effect on the affinity of the hemoglobin for oxygen and therefore affects how well the red blood cell function post transfusion. And this is leading us to look at the hemoglobin-oxygen dissociation curve. As we consider the hemoglobin-oxygen dissociation curve, let us remember that hemoglobin's primary function is to transport oxygen to the tissues and carbon dioxide, which is excretion, outside of the tissues and take it outside of the body. So one of the most important controlling factors in hemoglobin affinity for oxygen is red blood cell organic phosphate 2,3-DPG. So the uploading of oxygen by hemoglobin is accompanied by the winding of the spaces between the beta chains and the binding of the 2,3-DPG on the mole for mole basis, with the formation of an ionic salt bridge between these chains. The resulting formation of the deoxyhemoglobin molecule is known as TENS, or the T form, which has a lower affinity for oxygen. When the hemoglobin loads oxygen and becomes oxyhemoglobin, the established salt bridge are broken and the beta chains are pulled together, expelling the 2,3-DPG. This is the relaxed form or the R form of the hemoglobin molecule, which has a higher affinity to oxygen. These allosteric changes that occur as the hemoglobin loads and unloads oxygen are referred to as the respiratory movement. And I'm sure that you could have guessed that, because oxygen in, carbon dioxide out, and that there is respiration. We should realize by now that the dissociation and the binding of oxygen by hemoglobin are not directly proportional to the partial pressure of oxygen in the environment, but instead exhibit a sigmoid curve relationship known as the hemoglobin-oxygen dissociation curve. The shape of this curve is very important physiologically because it permits a considerable amount of oxygen to be delivered to the tissue with a small drop in oxygen tension. Where the PO2 tension measured in millimeters of mercury, mmHg, is near 100, the hemoglobin molecule is almost 100% saturated with oxygen. As the RBC travels to the tissue, where the PO2 drop to an average of 40 millimeter mercury, that's the venous oxygen tension, the hemoglobin saturation drops to approximately 75% saturation, releasing about 25% of the oxygen to the tissue. This is the normal situation of oxygen delivery at the basal metabolic rate. The normal position of the oxygen dissociation curve depends on three different ligands normally found within the red blood cell, which are the H plus ions, the CO2, and the organic phosphate. Of these three ligands, the 2,3-DPG plays the most important physiological role. Let us take, for example, a patient has hypoxia. If that is the case, there has to be a compensatory mechanism in the body, And this compensatory mechanism will promote or induce a shift to the right of the hemoglobin-oxygen dissociation curve, which alleviates the tissue oxygen deficit. The right shift of the curve mediated by increase of the 2,3-DPG, decreased hemoglobin affinity for the oxygen molecule, and increased oxygen delivery to the tissue. A shift to the left of the hemoglobin-oxygen dissociation curve will conversely result in an increase in the hemoglobin-oxygen affinity and a decrease in the oxygen delivery to the tissue. With such dissociation curve, the red blood cells are much less efficient because only 12% of the oxygen can be released to the tissue. Multiple transfusions of 2,3-DPG, depleted stored blood, can shift the oxygen dissociation curve to the left. So now that we understand about the basic mechanism of the blood and its operation, we need to understand now when the blood leaves the body, how is it that we are going to go about preserving it? So the goal of the blood preservation practice is basically to ensure the viability of the blood survival outside of the body. The US Food and Drug Administration, the FDA, requires an average of 24 hours post-transfusion red blood cell survival of more than 75%. Yes, 75% of the blood should survive. In addition, the FDA mandates that the red blood cell integrity be maintained throughout the shelf life of the stored red blood cell. This is assessed as free hemoglobin less than 1% of the total hemoglobin. So you know that with age, the cell may lyse, and lysine causes hemoglobin to be released. So they want to prevent the transfusion of lysed cells. Because if the cells have already been compromised and you're giving it to an individual who actually needs the oxygen carrying capacity of the red blood cell, then this is like giving someone something that is not effective or efficient. And we understand that the storage for long periods in some instances reduces the viability of the cells. In addition, the FDA mandates that the red blood cell integrity be maintained throughout the shelf life of the stored red blood cell. This is assessed as free hemoglobin less than 1% of the total hemoglobin. To maintain the optimum viability, blood is stored in a liquid state between 1 to 6 degrees for a specified number of days, as determined by the preservative used. The loss of red blood cell viability has been correlated with storage lesion, which is associated with various biochemical changes. As it relates to storage lesion, let us examine some of the things that are possible when this arise. So the viability of the cells, we are expecting that they will decrease. Glucose, based on extended storage, we're expecting that the cells will be utilizing the glucose that is available, hence that is going to be decreased. ATP is also a form of energy, so that there as well is going to decrease. Now let us move to the lactic acid. Lactic acid is normally built up when there is a stressful situation and oxygen is decreased. We expect the lactic acid to increase. And in this situation, we're expecting a change to be observed with an increased lactic acid. As it relates to the pH, if the lactic acid, which is increased, it means that there is a higher acidity. Hence, we are considering to have a decrease in the pH, because the lower the pH level, it means the closer it is to acidity. And now, when we examine the 2,3-DPG, we're also expecting that to be decrease based on storage, as we observed before, reduces 2,3-DPG, which is usually normalized after transfusion. Oxygen dissociative curve, we are expecting a shift to the left because increase in hemoglobin and oxygen affinity, and less oxygen is being delivered to the tissue because there is no tissue for the oxygen to be delivered to at this time. So we're expecting a shift to the left. Then we have plasma potassium. We're expecting plasma potassium to increase because when the cell integrity is compromised and there is a leakage of hemoglobin, then we have potassium seeping outside of the cell. Hence, we are expecting an increase. The plasma hemoglobin, we're also expecting that to increase because the potassium is increased. The plasma hemoglobin should correlate with also having an increase. So those just give us an idea of some of the storage lesions that we can expect. And as we stated before, the loss of red blood cell viability has been correlated with storage lesion, which means that the red blood cells are not able to function as effectively as they could prior. Low 2,3-DPG levels profoundly affects or influences the oxygen dissociation curve of hemoglobin. So DPG depleted red blood cell may have an impaired capacity to deliver oxygen to the tissue. As red blood cells are stored, 2,3-DPG levels decreases with a shift to the left of the hemoglobin-oxygen dissociation curve, and less oxygen is delivered to the tissue. It is well accepted, however, that 2,3-DPG is reformed in stored red blood cell after in vivo circulation. So once it's entered into the body, we're expecting that everything will be normalized, resulting in a restored oxygen delivery. The rate of restored 2,3-DPG is influenced by the acid-base status of the recipient, the phosphorus metabolism, the degree of the anemia, and the overall severity of the disorder. It has been reported that within the first hour after transfusion, most red blood cell clearance occur. Approximately 220 to 250 milligram of iron are contained in one red blood cell unit. Therefore, the rapid red blood cell clearance of even 25% of a single unit of blood delivers a massive load of hemoglobin ions to the monocytes and the macrophage system, potentially producing a harmful effect. So let us now examine some anticoagulant preservative solution for blood that is stored between 1 to 6 degrees Celsius. So some approved anticoagulant preservative solution includes acid citrate dextrose, which is formula A, that's ACD-A, and the storage time for this is 21 days. Then we have citrate phosphate dextrose, CPD, which is 21 days for storage. And then we have citrate phosphate double dextrose, which is also a 21-day storage period. But for citrate phosphate dextrose adenine, that's CPDA1, there is a 35-day storage time. We should make note that ACDA, acid citrate dextrose formula A, is used for apheresis components. Studies have shown that blood stored in all CPD preservative also becomes depleted of 2,3-DPG by the second week of storage. The report pathophysiological effect of the transfusion of red blood cell with low 2,3-DPG levels and increased affinity for oxygen includes an increase in the cardiac output and a decrease in the mixed venous PO2 tension, or a combination of these. Stored red blood cells regain the ability to synthesize 2,3-DPG after transfusion, but levels necessary for optimal hemoglobin oxygen delivery are not reached immediately. Approximately 24 hours are required to restore normal level of 2,3-DPG after transfusion. So the more age that the blood possesses, the higher the likelihood of a depletion of the 2,3-DPG. However, this can be rebalanced when transfusion takes place within a 24-hour period. But for patients which are normally the babies and also those with renal impairment, we tend to try to give them blood and blood products that are within 6 days old. We have also come to realize that many factors affect the viability of transfused red blood cell. One of these factors is the plastic material used for the storage container. The plastic must be sufficiently permeable to CO2 in order to maintain the high pH level during the storage. Now let us revisit some additive solutions. Additive solution AS are preservative solutions that are added to the red blood cell after removal of the plasma with or without platelets. Additive solutions are now widely used. There are 4 additive solutions that are licensed in the United States, and they are Adsol, that's AS1 from Fenwal Inc. Then you have Nutricel, that's AS3, and that is Hemonitrix Corporation. Then you have Optisol, that's AS5, and that is from Teramo Corporation. And then you have Solex, which is AS7, that is also from Hemonetics Corporation. And if you look at the denotations, they are all odd numbers 1, 3, 5, and 7. So 1 is Adsol. We realize that AS3 and AS7 are both from Hemonetics Corporation. What are some benefits, if there are any benefits, to using additive solution? Well, the benefit extends shelf life of the red blood cell to approximately 42 days by adding nutrients, allowing for the harvesting of more plasma and platelets from the unit. Produce a packed cell of lower viscosity, and this additive solution also produces packed cells of lower viscosity, which makes it easier to be infused. So all the additive solutions, AS1, 3, 5, and 7, they all have a storage time period of 42 days. And these are additives used in North America. For AS1, the storage time is 42 days, which is standard for all of the additives. The pH is 6.6. The survival after 24 hours is 83%. ATP is 68%. 2,3-DPG is 6%, and hemolysis is 0.5%. For AS3, storage time is 42 days, pH is 6.5%, 24 hours survival in percentage is 85.1%. ATP is 67%, 2,3-DPG is 6%, and hemolysis is 0.7%. AS5 storage time is 42 days, pH is 6.5. The survival after 24 hours is 80%. ATP is 68.5%. 2,3-DPG is 5%. And hemolysis is 0.6%. Now for AS7, the storage period is 42 days. pH is 6.6. 24-hour survival is 80%. ATP is 91%. 2,3-DPG is 1.5 micromoles per mil per gram of hemoglobin, and hemolysis is 0.3. Though we store blood at 1 to 6 degrees Celsius, blood can also be frozen. So we have a situation where we have freezing and rejuvenation of red blood cell. Red blood cell freezing is primarily used for autologous units and the storage of rare blood types. Autologous transfusion, or auto meaning self, allows an individual to donate blood for their own use to meet their needs for blood transfusion. And if you have a rare blood type and you know your family may need that blood, then you can donate over time. And this can be stored for up to 10 years. So basically, you're safe until it is needed. It involves the addition of cryoprotective agents to the red blood cell that are less than 6 days old, because after 6 days old, there is an increase in degradation of the blood cells, and you want to store blood at its highest real life viability. So glycerolized cells are utilized in that glycerol is used most commonly to be the additive which is infused with the blood by shaking, thereby enabling the glycerol to permeate the red blood cell. And this is usually stored at negative 65 degrees Celsius and below. The usual storage temperature is below negative 65 degrees, although storage and freezing temperature depend on the concentration of the glycerol used. So after this blood is stored, and 10 years later you will have to thaw it, or whenever it is needed it has to be thawed. Then there is something called the deglycerolization process which has to take place. This is a little technical and a little bit cumbersome. However, it serves a great purpose. It says transfusion of frozen cells must be preceded by a deglycerolization process. Otherwise, thawed cells would be accompanied by hypotonic glycerol when infused, and red blood cell lysis would occur. So in order to protect the viability of the blood itself and also the patient, to ensure that the blood is serving the purpose intended, it would be most prudent to remove that which was added so that the patient can get exactly what they need. With the glycerolization technique, there are two modes of operation. You have high glycerol and low glycerol. So they serve their own purpose in the different situations. So we're going to be looking at the initial freezing temperature, the need to control the freezing rate, a type of freezer, the maximum storage temperature, the shipping requirement, and the effect of change in storage temperature. So let us examine. So for high glycerolize, we need the initial freezing temperature of negative 80 degrees. There is no need to control the freezing rate. The type of freezer can be mechanical, and the maximum storage temperature after that temperature is reached is negative 65 degrees Celsius. Shipping requires only dry ice, and the effect of change in storage temperature can be thawed and then refrozen. For low glycerolize, the initial freezing temperature is negative 196 degrees Celsius. There is a need to control the freezing rate, and in this situation, the type of freezer you're going to use, you're going to have to use liquid nitrogen. The maximum storage temperature after the initial freezing temperature has been reached is negative 120 degrees Celsius. If it's going to be shipped, you're also going to need to ship it with liquid nitrogen in order to maintain that temperature. Effect of change in stored temperature is very critical. If the equipment that you have cannot reach below negative 80, then the high glycerol method is important. And if you don't want to control your freezing temperature, that is very important. And if you want to easily be able to ship it using dry ice, then high glycerol is important. Low glycerol would come in handy if you have a very controlled laboratory or research facility. Then you can just use your negative 96 degrees Celsius, use your liquid nitrogen, and do everything under controlled environment, and that would serve you well. So most persons, based on the requirements, opt into the high glycerol technique. Let us examine some advantages and disadvantages of actually freezing blood and blood components. So the advantage, number one, there's a long-term storage, usually 10 years, when the blood is frozen. Number two, there's a maintenance of red blood cell viability and function. Number three, there is low residual leukocytes and platelets. And number four, there is a removal of significant amount of plasma protein. So this is good if you just only want the blood. So some disadvantage, time-consuming process, that's number one. Number two, high cost of equipment and material. So those negative 80 and negative 190 degrees Celsius freezers, they are not cheap. Storage requires negative 60 degrees and a high cost of product. So these procedures will normally utilize in situation in which it's a research facility. It's an extremely rare blood type. The person is of prominence and great importance, and they can afford to deal with the costs associated with these procedures. But most facilities, they are not in a position to do that. So these will be left for institutions like schools and research facilities. Let us remind you that the rejuvenation process is expensive and time consuming. This is not used often. However, the process is invaluable for preserving selected autologous and rare units of blood for later use. Many persons may be considering, so if this is what they have done in the past, what are the current trends in blood banking in relation to red blood cell preservation, red blood cell utilization, red blood cell transfusion? Research is currently in development for red blood cell preparation and preservation, being pursued in 5 major areas. Development of improved additive solution. So they want to have solutions to extend the life of the blood products from 35 to 42 and from 42 to as long as they can. Now, there is also development of procedure to reduce and inactivate pathogens. The level of pathogen usually causes sepsis, thus reducing the viability of the blood and compromising the health of the person to which it has been transfused. There is currently development and procedure to convert A, B, and AB type red blood cell to O type, which is ongoing. There is also development of methods to produce red blood cell through bioengineering or blood farming. And there's also development for red blood cell substitution or having red blood cell substitute. These are currently ongoing and have not yet been FDA approved. So until then, we will just have to utilize the methodologies that are available to us. But as it relates to procedure to reduce and inactivate pathogen, a mucilline S303 pathogen reduction system is currently being studied and has demonstrated adequate post-transfusion viability according to the FDA criteria. As it relates to formation of O-type red blood cell, the enzymes are allegedly removed by washing after completion of the reaction time. As it relates to blood farming, in 2008, the Defense Advanced Research Project Agency, DARPA, awarded Arterioscience, a bioengineering company, a contract to develop a system for producing O-negative red blood cell on the battlefield. With the advent of the rise of HIV and transmitted blood diseases, there has been a call since the 1980s to find substitutes to alleviate the shortage of blood and to capitalize on the need for alternative and transfusion. Current research on blood substitutes is focusing on 2 areas: hemoglobin-based oxygen carriers, HBOCS, and perfluorocarbons, PFCS, originally developed to be used in trauma situations such as accident combats and surgery. Red blood cell substitutes have until recently fallen short of meeting the requirement of these applications. What could be some of the possible potential benefit of artificial oxygen carrier? So there is an abundant supply. It's readily available for use in hospital settings, battlefield, and remote location. It can be stockpiled for emergency and warfare. No need for typing and cross matching. It's available for immediate infusion. Extend the shelf life for 1 to 3 years. It can be stored at room temperature. It's free from blood-borne pathogen at full oxygen capacity immediately after transfusion. it does not prime circulation neutrophil, reduce the incidences of multi-organ failure. It can deliver oxygen to the tissue that is inaccessible to red blood cell. It have been accepted by Jehovah's Witness for those who are faith-based, and it could eventually cost less than a unit of blood because the testing would not have to be done like the HIV, HCLV, hepatitis B, antigen, hepatitis B, antibody, syphilis. Those testing would not have to be done. So we're going to look at some advantages and disadvantages of hemoglobin-based oxygen carriers. So the advantage of a hemoglobin-based oxygen carrier is that it has a long shelf life. It is very stable. There is no antigenicity unless bovine. There is no requirement for blood typing procedures. The disadvantage is that it has a short intravascular half-life, possible toxicity, increased O2 affinity, and increased oncotic effect. Now, let us just look at some advantages and disadvantages of perfluorochemicals. One, the advantage is that their biological inheritness, their lack of immunogenicity, and they're easily synthesized. The disadvantage of these are that there is adverse clinical effects, high O2 affinity, retention in tissue, and requirement for O2 administration when infused, not to mention the deep freeze storage temperature. So perfluorocarbons, the fluoro-DA, the first and only oxygen-carrying blood substitute ever to receive approval from the FDA for human clinical use in the United States, approved in 1989, but it was discontinued in 1994 because of clinical shortcomings and poor sales. Perforan, approved use in Russia and Mexico. PHER-O2, it's under evaluation for transfusion therapy for heart attack and stroke. This time, let us move our attention now to looking at platelets. Approximately 2.4 million platelets units are distributed, and 2.2 million platelet transfusions are administered yearly in the United States. With the limit of 5 days of storage for platelet concentrate, approximately 20 to 30% of the platelet inventory is discarded either by the blood supplier or the hospital's blood bank. So just like blood storage have red blood cell lesion, platelets also have their own sets of lesion. We call them the platelet storage lesion. Platelets are stored at 20 to 24 degrees Celsius with maintenance of continuous gentle agitation throughout the storage period of 5 days on an agitator. Agitation has been shown to facilitate oxygen transfer into the platelet bag and oxygen consumption by the platelets. That is one of the main reasons for platelets to be agitated, and not just only to prevent them from coming together. The positive role of oxygen has been associated with the maintenance of platelets components pH. Maintenance was determined to be a key parameter for retaining platelet viability in vivo when platelets were stored at 20 degrees Celsius to 24 degrees Celsius. The loss of platelets quality during storage is known as a platelet storage lesion. During storage, a varying degree of platelets activation occur that results in the release of some intracellular granules and a decline in the ATP and ADP. The reduced oxygen tension PO2 in the plastic platelet storage container results in an increase in the rate of glycolysis by platelets to compensate for the decrease in ATP regeneration from the oxidative TCA metabolism. This increases glucose consumption and causes an increase in lactic acid that must be buffered. This results in the fall in the pH. So we see the mechanism of action that is taking place. So during the storage of a platelet concentrate in plasma, the primary buffer is the bicarbonate. When the bicarbonate buffers are depleted during the platelet concentration storage, the pH rapidly falls to less than 6.2. And this fall to less than 6.2 is associated with the loss of platelet viability. In this state, the platelets will swell and there is a disc to spare transformation in the morphology that is associated with the loss of the membrane integrity. The platelets then become irreversibly swollen, aggregated together, or lyse, and when infused will not circulate or function. So it just becomes a waste. Generally, the quality control measures required by various accreditation organizations for platelet concentrates include platelet concentrate volume, platelet count, pH of the unit, and residual leukocyte count if claims of leukocyte reductions are made. Immediately before distribution to the hospital, a visual inspection is made that often includes an assessment of the platelet swirl and no visibility of aggregation. That is basically what we're examining. We don't want to see any discoloration, any white formation or mucoid formation within the platelet bag. The absence of platelet swirl is associated with the loss of the membrane integrity during the storage, resulting in the loss of discoid shape with irreversible sparing. So let us now examine the platelet storage lesion, the characteristics that are present and what we expect to observe, whether it increases or decreases. pH. The pH falls, so the pH will decrease. If the pH is decreased, we can assume the pH is decreased. The ATP over time will also decrease because that is energy. The lactate is going to increase, however. The morphological score changes from discoid to spherical, which means that there is a decrease in the viability. Degranulation is increased. Beta thromboglobulin, platelet factor 4, those are increased. Platelet activation marker, those would also be increased. That's P-selectin CD62P or CD63, that would increase. Platelet aggregation drop in response to some antagonists. So we're seeing a whole slew of problems taking place in this situation. In vitro platelet assay correlates with in vivo survival. So we think of the pH, the shape changes, the hypotonic shock response, the lactate production, and the PO2. Now, let us look at some of the value. 1 unit of whole blood derived platelet concentrate is supposed to be greater than 5.5 times 10 to the 10 platelets suspended in 40 to 70 mils of plasma, which is important. So 1 unit of whole blood derived platelet concentrate should be greater than 5.5 times 10 to the power of 10 platelets suspended in 40 to 70 mils of plasma. These plasma may be provided as a single unit or as a pooled unit. However, this is very important. If the units are pooled, then the shelf life now moves from whatever days it had left to 4 hours. That is now an open system. So to prevent or reduce bacterial contamination and to reduce any compromise of the platelets, then 4 hours after pooling, then it needs to be transfused. We're also observing that apheresis platelets contains greater than 3.0 times 10 to the 11, which is indicating to us that the apheresis techniques produce more platelets per unit, which is greater than 3.0 times 10 to the 11th power. So when it is derived from whole blood and it is manually done, extracted, then we have greater than 5.5 times 10 to the 10. However, when the machine does it, apheresis platelets are done, because it is more sensitive to the platelets, it can sieve specifically, so you have less passing out. So you have 3.0 greater than or equal to 3.0 times 10 to the 11 of 1 unit is the therapeutic equivalent to 4 to 6 units of whole blood derived platelets. So that there is a greater concentration of platelets made available from apheresis. Additive solution may be used for storage of apheresis platelets. In the United States, platelets are being stored in a 100% plasma media unless a platelet additive is used. Two platelet additive solutions are PAS, are FDA approved, and that's PASC, Intersol, and PASF, which is isoplate, for storage of apheresis platelets for 5 days. With the addition of PAS, residual plasma is reduced to 35% with both Intersol and isoplate. One advantage is that this approach provides more plasma for fractionation. Factors to consider when using a 5-day plastic storage bag for platelets. 1. The temperature control should be 20 to 24 degrees Celsius, which is typically room temperature. This is critical for both platelet preparation, platelet transportation, and platelet storage. Number 2, there should be careful handling of the plastic bag during the expression of platelet poor plasma, which helps to prevent the platelets bottom from being disturbed and prevent removal of excess platelets with the platelet poor plasma. Number 3, residual plasma volume recommended for storage of platelet concentrated from whole blood is 45 to 65 mils. Number 5, for apheresis platelets, the surface area of the stored bag needs to allow for the number of platelets to be stored. So now let us look at some advantage of using the platelet additive solution. 1, it optimizes platelet storage in vitro. 2, it saves plasma for other purposes, transfusion or fractionation. 3, it facilitates ABO incompatibility platelet transfusion. 4, it reduces plasma-associated transfusion side effects, such as febrile and allergic reaction, and may reduce the risk of transfusion-related acute lung injury. That's TRALI. Number 5, it improves effectiveness of photochemical pathogen reduction technology. Number 6, potentially improves bacterial detection. The reason why bacterial detection is so important with platelets is due to the temperature at which platelets are currently stored, which is 20 degrees Celsius to 24 degrees Celsius. And this is almost a sweet spot for bacterial growth, which is usually at 37 degrees Celsius. However, because of this storage technique, we have to be cognizant that bacterial growth is possible and probable. Hence, the preparation work needs to be done in terms of asking the client the pre-donor questions, also cleaning the area, and if possible, also running off the initial draw before collecting the final draw for the blood bag. That way you can reduce the potential of bacterial contamination of the platelet product. Environmental contamination during the processing and storage is another potential hazard that has to be tackled. Room temperature storage and the presence of oxygen provides a good environment for the bacterial proliferation. Sepsis due to contaminated platelets is the most common infection complication of transfusion. An estimated 10% of patients transfused with a bacterial contaminated platelet unit develop life-threatening sepsis, which is often fatal. So we have to be very vigilant as clinicians, whether medical technologists, laboratory scientists, or even the transfusion medicine practitioner. There are some techniques that are currently being used, like bacterial screening for platelets. However, they are not 100% sensitive. They may be supplemented by other precautions, such as the donor interview, a proper donor arm disinfection. Another precaution is the diversion of the first aliquot, about 20 to 30 mils of the collected blood, into a separate but contained diverse pouch. This procedure minimizes the placement of the skin plug, the most common source of the bacterial contamination. Traditionally, what happens after blood has been collected and platelets are paired prior to transfusion, about 4 to 6 platelet units are pooled together. And once this pooling take place, we know that we only have 4 hours before the units are no longer viable. In November 2009, the FDA approved the first rapid test to detect bacteria in platelets, the Pangenera Detection PGA test, Verax Biomedical. The PGA test, which was previously approved by the FDA for testing leukocyte-reduced platelets as an adjunct to culture, is an immunoassay that detects liponucleic acid on gram-positive bacteria and liposaccharide on gram-negative bacteria. Both aerobic and anaerobic are detected. A sample of only 500 microliters is required. So it has a built-in control that is run, and it turns from yellow to blue violet when the test is ready to be read. In approximately 20 minutes, a pink bar in either the gram-positive or the gram-negative test window indicates a positive result. The optimum time for sample is at least 72 hours after collection. But this test has its drawback and it has its problem. The rapid test has a sensitivity of 99.3% and a sensitivity of only 60%. While the rapid test is sensitive enough to detect majority of the platelets contaminating bacteria, a high false positive rate could lead to discarding of units of platelets that would be suitable for transfusion. The false positive rate of 0.51 with the PGE test is a valid concern because platelets are a blood product in short supply with a short shelf life. Despite sensitivity methods to detect bacteria in platelets, septic transfusion reactions still occur. So this area is currently ongoing as we don't have the bullet just yet to eliminate all the possible likelihood of platelet contamination. We should note that pathogen reduction for platelets procedures have been developed to treat platelet component to reduce or to inactivate any residual pathogens such as the bacteria, the virus, the parasite, and the protozoa that may be present. The FDA approved a pathogen reduction technology for apheresis platelets stored for up to 5 days as a measure to reduce the risk of bacterial contamination. The Intercept system, Soroscorp, use amoxicillin that is activated by ultraviolet A, UVA light, and binds to the nucleic acid-based pair of pathogen preventing replication. Dating of apheresis platelets up to 7 days is currently available in the United States. In order for blood centers to extend the date period from 5 to 7 days, the platelet must be collected in an FDA-approved 7-day platelet storage container. The 7-day container is labeled with a requirement to test every product with a bacterial detection device cleared by the FDA as a safe measured test. Number 2, the platelets are cultured at least 24 hours after collection with the FDA cleared devices and secondarily retest with the bacterial detection test labeled as a safe measure proximate to the time of transfusion. So these are some stipulation that needs to be undertaken if the lifespan of the blood is going to be extended. Let us just briefly examine some current trends in platelet preservation research. Research and development in platelet preservation is being pursued in many directions, including the following: development of better platelet additive solutions, development of more procedures to reduce and inactivate the level of pathogens that may be in the platelet unit. There's also development of platelet substitute. There is a new approach for storage of platelets at 1 to 6 degrees Celsius, similar to the storage of the unit of packed cell or whole blood. There's development of procedures to cryopreserve platelets. The idea of storing platelets at 1 to 6 degrees, this is not something that is new. This has happened in the past and it was discontinued many years ago. There has been an interest in developing ways to overcome the storage lesion that occurs at 1 to 6 degrees. So until that is done, platelets will continue to be stored at 20 to 24 degrees Celsius. Although considered for research techniques and not licensed by the FDA, frozen platelets are used occasionally in the United States as autologous transfusion for patients who are refractory to allogenic platelets. Platelets are collected by apheresis. The cryopreservative dimethyl sulfoxide, DMSO, is added, and the platelets are frozen at -80 degrees. The frozen platelets can be stored for up to 2 years. Prior to the transfusion, the platelets are thawed and centrifuged to remove the DMSO. Although in vivo recovery after transfusion is only about 33%, the platelet seems to function effectively. So now that we have discussed the erythrocytes and we have also discussed in part the thrombocytes, platelets, we have now concluded the basic fundamental concepts in understanding immunohematology, blood banking. And I hope that this information, this study session, this review has given you some insights into understanding not only what is on the paper, but using your mind to wrap around the concept so you can digest it and remember it as is appropriate. Red blood cells are erythrocytes and they serve the main function of carrying oxygen to the tissues and removing carbon dioxide from the tissues. The viability of these cells are important both within the body and outside of the body. When it comes on to transfusion and preparing for transfusion, we are reminded that we need 450 mils plus or minus 10%, and this is collected in 63 mils of anticoagulant. However, if it is 500 mils plus or minus 10%, then 70 mils of anticoagulants required. A donor can give blood every 8 weeks. Glycolysis generates 90% of the ATP and 10% is provided by the pentose phosphate pathway. 75% of post-transfusion survival of red blood cell is necessary for us to consider it a successful transfusion. Red blood cell is stored at 1 to 6 degrees Celsius for 21 to 35 days, depending on the anticoagulant or the preservative. The preservative that lasts for 35 days is the CPDA1. ACD, CPD, and the CP2D all last for 21 days. Additive solutions such as Addisol, Nutricel, Optisol, Solex are approved in the United States for red blood cell storage for up to 42 days. Red blood cells can be glycerized and frozen for up to 10 years. However, it has to be frozen within 6 days of collection, and it has to be collected in CPD or CPDA1. Rejuvisol is the only FDA-approved rejuvenation solution used to rejuvenate ATP and 2,3-DPG levels before freezing. In some cases, platelets can be stored for up to 7 days, but procedures have to be in place in order to secure that storage. Platelets can be frozen, and this is just experimental. This is not standard. When it is frozen, it can last for 2 years. So those are just some of the important information that we would have covered. In another sitting, we will present a recap or a refresh in a shortened format so that we can grasp some key clinical concepts. It was indeed a pleasure to share in these moments with you as we covered some of the fundamental concepts as it relates to red blood cells, erythrocytes, and the platelets. In our next sitting, we'll be doing some basic genetics, and then right after we'll move into the fundamentals of immunology so we can understand the key concepts of immunohematology. Thank you for stopping by, and feel free to share this information with your friends and your family who you know that may need a little assistance in understanding some concepts or would like to hear or the presentation from another individual to meet their learning need. If there is something that you'd like for us to do, please reach out to us and we'll try to make it possible. In another sitting, we'll be having some review questions where we'll be tackling these concepts which we have covered for your satisfaction. Thank you for enjoying with us the Professor Kareem Ainsley experience. Until next time, when we will continue our journey as we prepare for that which is to come.